Thank you, Anna, for the nice introduction. That helps with some of the slides that I'm going to show. I can refer back to the introduction. My name is John Bastiaansen. I work in Wageningen as a researcher on fish breeding programs and applications of genomics and research on genomics in several fish species. In Med8, we performed the we worked on the last objective that Anna showed on the list of objectives, which is to look at the genotype by environment interaction. And for this presentation, I gave a title that that's referring to the implications of this. So if we have genotype by environment interaction, does that mean that we can use the same broodstock or the same selection program for different environments? Okay, so I'm going to, I normally don't give you an outline, but it's a bit different from normal. So I'm going to talk about the objective and the rationale. So why is genotype by environment interaction important? What do we see as a result of in these interactions? And then I'm going to talk about a little bit about the experiment, how the data collection was done. Of course, present the results on G by E, genotype by environment interactions, conclusions, and after the conclusions, I still have a slide about implications. So what does that look like? This possible impact of G by E when you run a selection program. Okay, this is the objective as it was stated at the beginning of the project, and this is what we did. We assessed the existence of genotype by environment interaction. We aimed to vary the environments based on temperature. I'll show how that was done. And we aimed for traits that are related to production efficiency. And the way we measure genotype by environment interaction is by looking at correlations. And the first thing I'm going to do is to explain a little bit what these correlations look like. Anna explained that in a breeding program you have two groups of fish, and in this case they were also genotyped, like in that example. So parents, they produce a number of offspring that are grown in the test environment. Phenotypes, the traits are measured on these fish, and the genotypes are also taken on these fish. And then in genomic selection, you use these genotypes to select to predict the genetic value, the breeding values of the selection candidates, and select from the candidates. Now, what we did here is we had two test environments. So in this graph, we see I call them cold and warm. You see the two test environments, and in the Y direction, we see the breeding value, so the genetic value of an individual parent. So this parent had offspring in both environments, and that's why we can estimate its breeding value for both environments. And what you see is that this animal here at the top, it has two dots, one in the cold environment and one in the warm environment. And I connected them with a line to show how their ranking changes from one environment to the next. So you see that there's one here that's the top animal. It's excellent in both environments. But if you go down the list, you see that some of these lines, they have quite a slope. And the size of this slope is related to how sensitive the offspring of this parent are to the environmental difference. And that sensitivity varies. And that leads to a correlation that is less than 1 between the breeding values of parents for one environment and the other environment. So that's why we use a correlation to estimate the impact of genotype by environment interaction. A low correlation, meaning that it is close to 0 or not 1 and in the direction of 0, gives re-ranking of parents. So that has implications for selection depending on which production environment you're selecting for. So here you see I highlighted two individuals, two parents. The second from the top in the cold environment, it is number, I can't see it exactly, but maybe number 15 or 20 in the warm environment. And some other animals, they have a positive slope, so they would be better for the warm environment relatively to their ranking in the cold environment. It also means that it also means that if you do selection in one environment, so let's say we select in the cold environment, we would select the top animals on their breeding values. So this green accolade shows the group of animals that would be selected, and they would have a mean here somewhere in the middle. If you would produce their offspring in the warm environment, you see that there's more variation in the breeding values, and therefore the phenotypes, the performance of their offspring, but also that the mean performance in the other environment is lower. So the variation increases, but also the average goes down. So from selecting in the cold environment, you could expect a response based on this green accolade, and if you grow these fish in the warmer environment, you would see a lower average response. So it reduces the selection response in what I call the other environment. So if you have one selection environment, and production in the other, response in the other environment will be smaller. So what did we do in this experiment? We had a group of fish that were produced in Greece by project partner Galaxidi. They were spawned on a single day from a group of around 50 parents, so there were many families represented. And these fish were grown in the hatchery up to 3 grams, and at 3 grams they were stocked in a sea cage, both in Greece and in Spain. So we have this east-west, eastern-western Mediterranean distance between the production sites. We aimed to put in a contrast for the water temperature, and so that is shown here. If we look at the two grow-out locations, and on the y-axis here is the days in grow-out, and on the x-axis days in grow-out, and on the y-axis we have the cumulative temperature. So the degree days, the number of days times the average temperature that's accumulated over time. And we see that there is a difference in the pattern, and also, especially if you look at the end of the growing period, around 480 to 500 days, there is vertically quite a distance between Spain and Greece in the number of degree days. Also, along the growth trajectory, you see that the pattern in Spain is more smooth, so there's less sharp increases or decreases in temperature compared to Greece. So we have these not cold but warmer and less warm environments, and then at the end of the production period, we went to both sites, and we harvested from the sea case around 1,000 fish. We phenotyped them for the traits that are here listed on the right. We have harvest weight, fillet weight, visceral weight, and fillet fat. Here at the top in the picture, you see some of the students, masters and PhD students that were involved in the practice. You see Yun, Varun, and Xiaofai working on measuring fat percentage with the Distel equipment and taking the fin clip samples to be genotyped with the medfish array. And like we said in the objective, we measured G by E by estimating the genetic correlation between the sites for the breeding values of these traits. So what did we get? So we get a single number. We get a genetic correlation value, which are listed here. And we see that for let's say the growth, production related traits, harvest weight, growth rate, and fillet weight, genetic correlations are in the 0.45 to 0.5 region, which is quite low. For comparison, the graph that I was showing earlier, which you see here on the right, that's for a correlation that's about 0.7. So with 0.7, you already get the re-ranking that we see here on the right. And in the experiment, we found that the genetic correlations are still a bit lower than what you see here on the right. So quite low for the production traits in the two environments. We also saw that for fillet fat, we have a quite high correlation, 0.87, so almost 0.9, which means that the genetic value for a parent to produce fillet fat, for fillet fat levels in Spain versus Greece are quite similar. So if you select in one environment, you expect a similar response also in the other environment. Okay, so the first set of conclusions is that between the Eastern and the Western Mediterranean sites, so selected for colder, warmer and less warm temperatures. Of course, there are other differences between sites that contribute, but the experiment is designed to specifically target the temperature. And we see strong G/E for these production traits, harvest weight, fillet weight, and growth rate in gilthead seabream. So this, the conclusions are specific to the species, of course, that is tested. We see very limited, or maybe no, given the standard error, maybe no genotype by environment interaction for fillet fat percentage. And also, I didn't show the values, but the heritabilities of all these traits are moderate to high. So they're all between the 0.3 to 0.5 even, which means that 30% to 50% of the variation that's observed is due to genetic differences between individuals. And that gives an indication that selection for those traits should be quite effective. And of course, that's also what has been seen in breeding programs for these species. Then coming back to the title, can we supply all these sites from one broodstock, or let's say broodstock from a single breeding program? Given the rather low genetic correlations, we conclude that you cannot simply select in one environment and expect that selection to lead to the response that you predict in the other environment. So you need to take into account which production environment you are targeting. So if you're doing a selection program for a specific production environment, you test your fish in that environment. If you have a breeding program and you supply to multiple diverse production locations, then you will need to think about collecting data for your selection in those, in a subset at least of those multiple locations where the fish from your breeding program are expected to produce. We specifically targeted temperature also to be able to say something about the future. Breeding is a long-term endeavor, so it's breeding is always for the future, we say. So if in the future you expect changing conditions, like increasing temperatures due to climate change are an issue already, so then it is important to take this also into account when you're breeding fish for the future. So if you expect raising higher temperatures, then you emphasize more the traits that you're measuring in the higher temperature environment. So that could future-proof your breeding program by using those data points from locations that look more like future conditions, maybe in other parts of the Mediterranean as well. Okay, then the one before last slide. So what does it mean? So in quantitatively, so how does it affect what we do in breeding programs, or what should be done in breeding programs? So breeding programs aim to get a response to selection, to improve the population. And I put a little bit of quantitative genetics formula here. say one of the most important formulas, is the response that you get depends on three things. How intense you select, so how much better your selected animals are than the average. The accuracy, I put that here in red because the correlation relates to this, and how much variation there is. So if you take the top 10% from a very narrow variance, you don't move as much as when you take the top 10% from a very wide variance in your population. But this genetic correlation between environments, it affects the accuracy. So if you select in one environment, that measurement in that one environment has a reduced accuracy for improvement in the other environment. And with the values that we estimated in this experiment, I show in these bottom two graphs on the left the response in harvest weight. So I put time on the x-axis and genetic level on the y-axis. I didn't put a scale there because it depends. The scale also depends on the other two parameters in this equation, intensity and variance. But for a given intensity and variance, the picture looks like this. So this green line, it shows selecting in one environment and the response that you would expect in that environment. Now, if you then move those fish to the other environment, the response over time, so the genetic improvement over time that you observe, is shown by this red line. And you see that there's about a 50% reduction, which is close to this value of these values of the genetic correlations. Now, in comparison, if you do the same exercise for fillet fat, you see that the responses in the selection environment and in the targeted environment are very similar. So for a trait like that, or in a situation like that, you are okay with using the breeding program in one environment to see improvement in all of the production environments. Well, the ones that are tested in this experiment. Okay, this is the final slide. Thank you to everybody involved in the experiment. There's a lot of names here, a lot of students that participated, collaborators. Of course, people in the MedAid consortium that made everything possible. And a big thank you to the companies that hosted us, that hosted the fish, and that also hosted us to come and do the phenotyping. Thanks a lot. We had a good time, and also we have very good results from our experiments.
Mm-hmm. Okay.
Thank you.
Thank you very much, John.